Swayam Prabha. Digital India. Educated India.
Hi students, I am Dr. Kalpana Ramachandran. I am Professor and Head, Department of Anatomy, Sri Muthukumaran Medical College, Hospital and Research Institute, Chennai. Today I will be giving a lecture on the microscopic anatomy of kidney. Let us begin this lecture with the functions of kidney. The main function of kidney is maintenance of homeostasis by filtration, active absorption, passive absorption and secretion. So, what is urine? Urine is plasma which has been modified by passing along the tubules. To understand the microscopic anatomy of kidney, let us learn about the coronal section of kidney, which is the gross anatomy of kidney, for better understanding of the microscopic anatomy. So, kidney on a coronal section, as you can see from this picture, has a outer cortex and a inner medulla which consists of cone shaped structures which are called as renal pyramids. These cone shaped structures are called as renal pyramids. The pyramids have a base which is at the corticomedullary junction. The pyramids have an apex which is going to project into the minor calyces which are funnel shaped. As you can see from this picture, these minor calyces are going to join and they form the major calyx. The major calyces join and form the funnel shaped renal pelvis. So, the renal pelvis is going to emerge through the hilum of kidney. This is the hilum of kidney through which the renal pelvis is going to emerge out and is going to continue as the narrow muscular ureter. Now, we will go into the renal columns of Burton. Renal columns of Burton are nothing but extension of the cortical tissue in between the pyramids of medulla. These are all the important gross anatomical structures which are very relevant to the study of microscopic anatomy. This is a histological picture of the kidney. So, kidney in a histological picture is divided into an outer dark staining cortex and inner pale staining medulla. So, this is cortex and this one is the medulla. Externally, cortex is covered by a dense irregular connective tissue capsule. So, what are the structures which are seen in the cortex? So, cortex consists of renal corpuscles, proximal convoluted tubule, distal convoluted tubule and medullary rays. You can see these are the medullary rays. So, what are they? They are longitudinal striations in cortex which are going to course towards the medulla. This is the medulla of kidney which contains collecting tubules, collecting ducts and segments of loop of Henle. This is a low power view of the kidney. Let us look at a slightly enlarged view, that is a high power view of kidney, to look at the medullary rays. So, you can see this is the cortex of kidney where you can see the medullary rays. As I told you, they are longitudinal striations in the cortex which course towards the medulla. I will give the details of medullary rays as we proceed further. Before going into the histological structure of each of the structures in the cortex and medulla, let us first define what is uriniferous tubule. So, the functional unit of kidney is uriniferous tubule. As you can see from this picture, the uriniferous tubule consists of nephron and collecting duct. Uriniferous tubule is nephron and collecting duct. So, nephron consists of renal corpuscle and renal tubules. So, nephron and collecting duct together are called as uriniferous tubules. Again, the uriniferous tubules can be divided into two parts, an secretory part and a collecting part. Excretory or secretory part is the nephron that produces urine. Excretory or collecting part is the collecting duct which is going to conduct urine. Let us look at some of the details of the uriniferous tubule. As I told you, the uriniferous tubule has got nephron and collecting duct. So, nephron consists of a dilated part which is called as renal corpuscle. This is renal corpuscle. What is it for? It is for filtration from plasma. And the tubules are for selective reabsorption. These are the two parts of the nephron, dilated renal corpuscle and tubules. So, the renal corpuscle, if you take, has got again two parts. It has got glomerulus, which is a tuft of capillaries, and outermost is the Bowman's capsule. So, renal corpuscle consists of a Bowman's capsule and a glomerulus, which is nothing but tuft of capillaries. Now, we will go into the tubules. What are the various parts of the tubules? As you can see from this slide, the tubules consist of proximal tubule, Henle's loop, distal tubule and collecting tubule. The four parts again I repeat are proximal tubule, Henle's loop, distal tubule and collecting tubule. Let us position each of these in a section of kidney, in the cortex and in the medulla. So, proximal tubule is solely seen in the cortex. This is the cortex, this is the medulla. Proximal tubule is solely seen in the cortex. It begins from the renal corpuscle. It has got a convoluted part which is in the cortex, that is called as pars convoluta. It has got a distal part which is called as the pars recta of the proximal convoluted tubule, which is also called as the thick descending limb of loop of Henle. So, proximal tubule has got pars convoluta solely in the cortex and a straight part pars recta of proximal convoluted tubule, which descends from the cortex into the medulla. The Henle's loop has got four parts. One is the pars recta of PCT, which is the thick descending limb. Then you have a thin descending limb. Then you have a thin ascending limb. These are seen in the medulla. Then you have a straight part or pars recta of distal convoluted tubule, which is nothing but the thick ascending limb of loop of Henle. As you can see from this picture, the thick ascending limb from the medulla courses towards the cortex and then becomes more convoluted and forms the convoluted part of the distal tubule. So, distal tubule has got a convoluted part which is called pars convoluta and a straight part which is called as the pars recta of distal convoluted tubule, which is nothing but the thick ascending limb of loop of Henle. The distal convoluted tubule connects with the collecting tubule. You can see in this picture, this is outer cortex, this is the inner medulla, the distal convoluted tubule is going to join the collecting tubule. So, you can see that number of collecting tubules from different nephrons are going to join and form the collecting duct. This is a large collecting duct which courses from the cortex towards the medulla. So, it is descending towards the papilla of medulla. So, these ducts, large ducts formed by union of collecting ducts is called as the papillary ducts. Papillary ducts are also called as ducts of Bellini. So, this is how the arrangement of various structures in the cortex and medulla is present. So, you can see that in cortex you have renal corpuscles, you have PCT, DCT and medullary rays. Medulla, you have collecting tubules, collecting ducts and you have segments of loop of Henle. So, let us look at what are medullary rays. These medullary rays, as I already told you, are longitudinal striations which are seen in the cortex, which are going to extend down into the medulla. So, this medullary rays contains blood vessels, they contain straight part of PCT, that is the thick descending limb of loop of Henle, straight part of distal convoluted tubule, that is the thick ascending limb of loop of Henle, collecting tubules and collecting ducts. Let us look at the types of nephrons. There are two types of nephrons based on their position. One is the cortical nephron, which is again divided into superficial and mid cortical depending upon their position. Please note, the cortical nephrons, their tubules do not extend deep into the medulla. Cortical nephrons, tubules of them do not extend deep into the medulla. Second one is the juxtamedullary nephrons. This is the juxtamedullary nephrons which are the largest. Their tubules extend deep into the medulla. Having talked about the various structures which are present in the cortex and medulla, before going into the histological details of these structures, there are two things which are very relevant when you study the histology of kidney. One is a renal lobe, the other one is a renal lobule. Let us go on to define renal lobe and a renal lobule. What is renal lobe? Please look at this picture. The renal tissue in the medullary pyramid, this is the medullary pyramid with its cap of cortical tissue, whose collecting duct is going to enter the pyramid is called as renal lobe. What is shown between these two lines is the renal lobe. This is the medullary pyramid with its cap of cortical tissue. The collecting ducts enter this pyramid. So, that is renal lobe. Next is renal lobule. You can see from this picture that renal lobule is a portion of the renal cortex. This is cortex, this is medulla. So, renal lobule is portion of renal cortex draining into a single collecting duct, that is the renal lobule. So, now we will summarize what we have learned till now, the general arrangement of structures in a histological section of kidney. As I have already told you, kidney is divided into an outer darker staining cortex and an inner pale staining medulla. There is a capsule covering the cortex. Cortex has got renal corpuscles, PCT, DCT and medullary rays. Medulla consists of collecting tubules, collecting ducts and segments of loop of Henle. This is the picture showing the outer cortex and inner medulla to show the general arrangement of structures in the cortex and medulla of kidney. As I told you, the cortex contains these structures and medulla contains collecting tubules, collecting ducts and segments of loop of Henle. This is repeated because we are going to study in detail the histological structure of each of these structures in the cortex and medulla. Now, we will first take renal corpuscle, or it is called as Malpighian corpuscle, the structure of which will be detailed in the next few slides. So, renal corpuscle, as I told you, consists of Bowman's capsule and a glomerulus. So, this renal corpuscle is 200 micrometer in diameter. So, glomerulus is nothing but a tuft of capillaries. How many tufts? 10 to 12 loops of capillaries are present in it. As you can see from this diagram, this renal corpuscle is supplied by an afferent arteriole and it is drained by an efferent arteriole. So, little more details about the renal corpuscle. The Bowman's capsule has got two layers. The outer layer is the parietal layer and the inner layer which embraces or envelops the capillaries of the glomerulus is called visceral layer. So, this is parietal layer, outer layer and inner visceral layer which envelops the capillaries. So, there is a space between these two layers that is called as urinary space, what I am pointing out, or it is also called as Bowman's space or capsular space. So, what is this Bowman's space continuous with? You can see from this diagram that the Bowman's space is continuous with the lumen of proximal convoluted tubule. So, this each renal corpuscle has got two poles. One is the vascular pole. How to identify vascular pole? Vessels enter and leave, that is afferent and efferent arterioles are present in the vascular pole. The opposite pole is the urinary pole. You can see that PCT begins here. So, there are two poles for the renal corpuscle, vascular pole and a urinary pole. Let us look at the histological details of renal corpuscle. The parietal layer of Bowman's capsule, as you can see this histological section, is lined by simple squamous epithelium. This is simple squamous epithelium. The cells of the visceral layer which are going to embrace the capillaries, they are called as podocytes. The cells of the visceral layer which embrace the capillaries are called as podocytes. This podocyte requires little more detailing, which we will be seeing in the next slide. So, the podocyte, as I told you, has got a cell body. This is the cell body from which arises major processes. These are the major processes, or they are called primary processes. From the primary process, you have secondary process or pedicels or food processes. What you see in this diagram is the cell body, primary process and secondary process or food process or pedicels. Food process or pedicels of the podocyte which is coming in contact with the capillary. So, at periodic distance, the pedicels are going to embrace the capillaries of the glomerulus. They are going to interdigitate with the pedicels of the adjacent podocyte. This is one podocyte, this is other podocyte, this is the pedicel of this podocyte and this is the pedicel of the adjacent podocyte. So, they interdigitate with the pedicels of adjacent podocyte. So, between adjacent food processes you have gaps. These are the gaps which are seen, which are called as filtration slits, through which filtration is going to occur. They are called as filtration slits, or they are called as pores, filtration pores. So, these filtration slits are closed by a diaphragm which is called as slit diaphragm. Why do you need to know the details of podocytes, the filtration slits and other things what I have detailed? It is because these filtration slits what you are seeing in this diagram act as mechanical barriers. They act as mechanical barriers. They permit water, glucose and amino acids through the filtration slit, but they prevent large protein molecules such as albumin from passing through. That is why the histological significance of these structures are to be noted. These filtration slits act as mechanical barriers. So, we have been talking about a filtration slit, pore. What is the significance of this? There is a barrier which is called as filtration barrier between the food process of podocyte, this is the podocyte, the food process, and the glomerular basement membrane through which filtration occurs. What is this glomerular basement membrane? It is nothing but fused basal lamina of food process and the basal lamina of the endothelium. Third thing which constitute the filtration barrier is the endothelium which is fenestrated, the endothelium of the capillaries. So, three things constitute the filtration barrier. One is the food process of podocyte, second is the glomerular basement membrane, third is the endothelium of the capillaries which is fenestrated, through which filtration is going to occur. You see in this picture, filtration will occur through the filtration barrier. So, the glomerular filtrate if you take has got a chemical composition which is similar to plasma, but please note almost no proteins. It is similar to plasma, but almost no proteins, as you can understand that the filtration slits act as mechanical barriers and they do not allow proteins to pass. So, that is about the renal corpuscle. In addition to this, you have certain cells called as mesangial cells between the glomerular capillaries. Why do you need to know about this mesangial cell? It is a very important cell which provides structural support, and one more important function is they phagocytose the trapped immune complexes. That is about the mesangial cell. Having studied about the renal corpuscle, the detailed histological structure, we will move into the next structure, that is the proximal convoluted tubule. The proximal convoluted tubule, the convoluted part is solely in the cortex, and the distal part which we called it as pars recta of PCT, which is also given another name, that is the thick descending limb of loop of Henle, which is going to descend from the cortex into the medulla. So, now we are going to see the histological feature of proximal convoluted tubule and also the pars recta of PCT. So, if you take a section of the cortex, what you are seeing here is the renal corpuscle, and you can see many cut sections of tubules. So, in the cortex you will be seeing renal corpuscles, PCT, DCT. In a section of the cortex, you will see more sections of PCT than DCT. This is a very important point you must note. There are more sections of PCT than DCT. Why is it so? It is because PCT is the longest and widest part of the nephron, and it is going to form the bulk of the cortex. So, you will see more sections of PCT as compared to that of the DCT. So, in the histological section, how do you identify proximal convoluted tubule? This is one proximal convoluted tubule which is pointed out through an arrow. You can see that it is lined by cuboidal cells. They are lined by cuboidal cells. The nuclei is spherical. It is eosinophilic, darkly stained eosinophilic, that is because you have high content of organelles, that is mitochondria. Also from the apical surface of the cell you have tall microvilli. Tall microvilli is seen, which is called as brush border. Let us have a closer look at the proximal convoluted tubule. I told there is a brush border which is nothing but the tall microvilli which are irregularly arranged. So, this is the proximal convoluted tubule. As I told you, the apical surface has got microvilli. Why do you need microvilli on the apical surface? This is to increase the surface area by 20-fold. It increases the surface area by 20-fold. There are also apical canaliculi which are nothing but tubular invaginations between the microvilli. So, there is importance for this apical canaliculi also. They endocytose molecules which have crossed the glomerular filtrate. The function is endocytosis. Also in the cell, cuboidal cell, you can see some basal infoldings in between or mitochondria which are present. These are called as basal striations. Basal striations in the cell of the PCT. Also lateral cell boundaries interdigitate, that means cell margins are not clearly definable. So, this basolateral plasma membrane of the cell has got a pump which is called as sodium potassium ATPase pump, which is responsible for reabsorption of sodium. This is very important. The pump is present, the mitochondria present here provides energy for the pump. This pump is responsible for reabsorption of sodium. Water and other solutes pass easily. So, the proximal convoluted tubule reduces the glomerular filtrate by 80 to 85%. This is because of its histological structure. The proximal convoluted tubule, I repeat, reduces the glomerular filtrate by 80 to 85%. So, now we will summarize what we have learnt till now. We have mostly concentrated on the cortex of the kidney. We have learnt in detail about the renal corpuscle, which consists of the Bowman's capsule which is outer and glomerulus which is nothing but tuft of capillaries. Then we have talked about the proximal convoluted tubule in detail, and please understand that there are more sections of PCT as compared to DCT in the cortex because PCT is longest, forms the bulk of the cortex. They are darker staining with tall microvilli which are forming the brush border. So, lumen is not clearly defined in a PCT. Now, let us move on to the next segment of the tubules, that is the loop of Henle. So, as already detailed, there are 4 parts: thick descending limb of loop of Henle, thin descending limb, thin ascending limb and thick ascending limb of loop of Henle, which is nothing but pars recta of distal tubule. The lengths vary based on the position of the nephron. We will look at the histological structure of thick descending limb. You can see in this histological picture the thick descending limb of loop of Henle, which is very similar to proximal convoluted tubule. But there are few differences. One is that the thick descending limb is shorter, few mitochondria are present and less basal striations are present. So, in the outer medulla it changes into the thick descending limb changes into thin segment. So, how to identify thin segment of loop of Henle in the medulla of the kidney? The lining epithelium is, you can see from this structure which is pointed out, which is nothing but thin segment of loop of Henle. The lining epithelium is simple squamous epithelium. You can see that nuclei protrude into the lumen. So, there comes the question how to differentiate a capillary from a thin segment. Capillaries have in their lumen erythrocytes, but you do not find erythrocytes in the lumen of thin segment, and capillaries the epithelium is thinner, but you can see that here the epithelium is thicker and the nuclei protrude into the lumen, that is thin segment of loop of Henle. Thick ascending limb is lined with cuboidal cells like thick descending limb, it is more rounded. You can see from this picture it is more rounded, but please note there is no apical microvilli, that is brush border. So, lumen is clearly defined or seen. So, what is the function of loop of Henle? Before we move on to the next part of the tubule, we will talk about the loop of Henle. The descending limb of loop of Henle is permeable to water, but not solutes. The ascending limb of loop of Henle is permeable to solutes, there is an active pump, but not to water. So, you can note that there is an increasing osmotic gradient within the renal interstitium and vasa recta. Vasa recta are nothing but parallel wide capillary loops which lie parallel to the loop of Henle. So, this increasing osmotic gradient is going to facilitate the counter current mechanism of reabsorption. Counter current mechanism of reabsorption. I have told you what are the structures, what are the substances which are reabsorbed in the descending and ascending limb of loop of Henle. Now, we will come to the distal tubule. So, we have talked about the distal tubule having two parts. One is the thick ascending limb of loop of Henle, which is the pars recta of distal tubule, which is going to ascend from the medulla, reaches the cortex. Please note here that the first part of this, once it returns to the vascular pole of the parent nephron, this is the thick segment of the loop of Henle, ascending to the cortex, and it reaches the vascular pole of the parent nephron. It forms a structure called as macula densa of juxtaglomerular apparatus. I will be talking in detail about the juxtaglomerular apparatus. Understand that once the ascending limb ascends to the cortex, it reaches near the vascular pole of the renal corpuscle of its parent nephron. The first part is going to form the macula densa. Beyond this landmark, it is going to form the distal convoluted tubule. We will now look at the structure of a distal convoluted tubule. This is a section through the cortex, a high power view. You can see so many sections of proximal convoluted tubule as compared to distal convoluted tubule. You know the reason why it is like that. So, in the histological section, how to identify distal convoluted tubule? The distal convoluted tubules are also lined by cuboidal cells. You can see from this picture, but they are paler staining as compared to the PCT, which is dark staining. They have spherical nuclei. There is no brush border, so the lumen is well defined. Compare this PCT with DCT. Lumen is very well defined because there is no brush border. The cells are smaller, so more nuclei per section are seen. Have basal striations. What is the function of distal convoluted tubule? They selectively reabsorb sodium and excrete potassium In exchange, the DCT is under the influence of antidiuretic hormone and aldosterone. This is very, very important. DCT is under the control of ADH and aldosterone, and so it selectively reabsorbs sodium and excretes potassium. Having read about PCT and DCT, let us state the differences between PCT and DCT as a summary. This is PCT, what is pointed out on the left. On the right is a DCT. PCT is longer, so more sections of PCT are seen. DCT is shorter, so you will see fewer sections of DCT. Dark stained is a PCT, light stained is a DCT. PCT has got large cells, so smaller lumen, fewer nuclei because you have large cells. DCT cells are smaller, as I have already mentioned, larger lumen, more nuclei. You can see that there are more nuclei per section, it is around 6 to 8. Since there is brush border here, lumen is not well defined, but in DCT lumen is well defined because there is no brush border. So, now we will go into the next histological aspect of kidney, which is very, very important in regulation of blood pressure, that is the juxtaglomerular apparatus. So, this juxtaglomerular apparatus has got various components. First is the macula densa. As I have already mentioned, this is the renal corpuscle with its afferent arteriole and efferent arteriole, and this is the distal convoluted tubule, which is establishing contact with the vascular pole of the renal corpuscle of its parent nephron. This is DCT, this is a parent renal corpuscle. So, it is establishing contact with the vascular pole of the renal corpuscle of its parent nephron. So, the cells in contact with the afferent arteriole, this is afferent arteriole, become columnar in the wall of the distal convoluted tubule. The nuclei become closely packed. This is called as macula densa. This is nothing but the distal tubule, the cells in contact with afferent arteriole become columnar and nuclei become closely packed, that is your macula densa. What is the importance of this? They are sensors of fluid in the distal tubule, and so they will stimulate cells which are called as juxtaglomerular cells. So, what are these juxtaglomerular cells? These juxtaglomerular cells are seen in the wall of the afferent arteriole. Look at the picture. They are modified smooth muscle cells in the tunica media of afferent arteriole. The cytoplasm is going to be packed with secretory granules. This JG cells or juxtaglomerular cells respond to changes in blood volume and they are stimulated by the macula densa. See the close association between macula densa and juxtaglomerular cells. These two are in close association. So, what is juxtaglomerular apparatus? As you can see from this picture, it has got three components. One is macula densa in the distal tubule, two is juxtaglomerular cells in the wall of afferent arteriole, they are the modified smooth muscle cells. And third component you can see from this picture, lying between the afferent and efferent arteriole is the pale staining cells called as Lassus cells. Lassus cells, they are given different names. They are extraglomerular mesangial cells, they are also called as Pulkyson cells, or they are also called as Gourmatic cells. They are going to lie between the afferent and efferent arteriole. Why do you need these cells, Lassus cells? Please note the importance of these cells. They will transmit impulses from the macula densa to the JG cells. You can understand that there are three components of juxtaglomerular apparatus: macula densa, juxtaglomerular cells, and a cell which is laces cells which lie between afferent and efferent arteriole. They transmit impulses from macula densa to the juxtaglomerular cells. Having understood the histological structure of the juxtaglomerular apparatus, now I am going to tell you the role of juxtaglomerular apparatus in control of BP. Why should you read in detail about the juxtaglomerular apparatus, its histological structure? It is because juxtaglomerular apparatus is going to serve as both a baroreceptor and as a chemoreceptor. Let us look at the role of JGA in control of blood pressure. So, whenever there is going to be a fall in the blood pressure, the amount of glomerular filtrate which is going to reach the distal convoluted tubule is going to be very less. Once the filtrate is very less, the amount of sodium in the filtrate is also less. This is sensed by the macula densa. I told you in the distal convoluted tubule there are cells which are modified, the cuboidal cells are modified to become columnar and they are called as macula densa. So, these macula densa sense that there is less amount of glomerular filtrate coming, there is less amount of sodium, and what they do is they immediately stimulate the adjacent juxtaglomerular cells. They stimulate the juxtaglomerular cells to produce the enzyme renin. So, this enzyme renin is going to enter the circulation, and what it does is this enzyme renin converts angiotensinogen, which is nothing but a plasma protein, into angiotensin 1. This angiotensin 1 is converted in the lung by angiotensin converting enzyme into angiotensin 2. Please note this angiotensin 2 is the one which is going to increase the blood volume and blood pressure. We have already told you that the blood pressure has fallen, less glomerular filtrate. So, the blood pressure has to increase, the blood volume has to increase. So, this occurs because of renin secretion, conversion of angiotensinogen to angiotensin 1 into angiotensin 2. So, this angiotensin 2 acts by two ways. One is by vasoconstriction that results in increased blood volume and blood pressure. Second is it stimulates the adrenal cortex to produce aldosterone. Stimulates the adrenal cortex to produce aldosterone. As you all know, aldosterone will cause increased water and sodium absorption, reabsorption from the distal convoluted tubule. Again, this in turn increases the blood volume and blood pressure. So, whenever the blood pressure falls, the juxtaglomerular apparatus is going to be in action, and that results in increase in blood volume and blood pressure. Having learnt about various structures in the cortex and medulla, we will talk about now about the collecting tubules and the collecting ducts. The distal convoluted tubule drains into the collecting tubules, they drain into the collecting ducts, which run in the cortex, that is in the medullary rays, enter the medulla. These collecting ducts join to form a large duct which is called as the duct of Bellini. So, what is the histological structure of collecting tubule and collecting duct? You can see from this picture that the collecting tubules are wider, they are lined by pale staining cells, low cuboidal cells. So, wider, low cuboidal cells line the collecting tubule and they are pale staining. The cell margins are well defined. The epithelium increases in height as it reaches the inner medulla, the collecting tubules. The collecting ducts have a larger diameter and they have a columnar epithelium. Collecting ducts drain into the duct of Bellini. You can see that duct of Bellini is lined by simple columnar epithelium. So, this collecting ducts of Bellini are going to pierce the papilla, renal papilla of the pyramid at the lamina cribrosa. This is the area of lamina cribrosa, and drain into the funnel shaped minor calyx. We have started from structures in the cortex, then medullary pyramid, and then now we have come to the collecting ducts which of Bellini, which pierce the papilla and they open into minor calyx. The collecting system from minor calyx, that is minor calyx, major calyx, renal pelvis, all are lined by transitional epithelium, which is also called as urothelium. Transitional epithelium is also called as urothelium. So, what are the structures seen in renal medulla? Renal medulla consists of collecting tubules, collecting ducts, thick and thin segments of loop of Henle and blood vessels. The histological structure of all these tubules and ducts and segments of loop of Henle have been highlighted in the previous slides. Finally, is the renal interstitium. What is renal interstitium? It is a connective tissue which is in the kidney. The connective tissue is sparse in the cortex, but it is more abundant in the medulla as compared to cortex. This is the renal interstitium which you see. It is composed of matrix of highly hydrated proteoglycans, collagen and reticular fibers and connective tissue cells. So, having learnt about the various histological structures in the section of kidney, let us summarize what we have learnt in a histological section of kidney. As you can see from this picture, kidney is divided into outer cortex and inner medulla. We have seen that the functional unit of kidney is uriniferous tubule, which consists of nephron and collecting duct. Nephron is nothing but renal corpuscles and renal tubules. As you can see from this picture, the cortex consists of renal corpuscle. Outer to this is the proximal convoluted tubule, distal convoluted tubule and medullary rays. The medulla consists of collecting tubules, collecting ducts and segments of loop of Henle. We have seen the structure of detailed structure of PCT and DCT as seen in the cortex. I have already mentioned that PCT is longer, forms the bulk of cortex. So, you will see more sections of PCT as compared to DCT. PCT is darker staining, DCT is lighter staining. PCT has got brush border. So, lumen is not well defined. Here there is no brush border and hence lumen is very well defined. Less number of nuclei because there are large cells in PCT, more nuclei of the DCT because you have smaller cells. So, this is PCT as compared to DCT, and this one is the renal corpuscle as seen in the cortex with its outer Bowman's capsule and inner glomerulus, which is nothing but tuft of capillaries. We have also detailed about the juxtaglomerular apparatus, which includes three structures. One is macula densa in the distal tubule, juxtaglomerular cells in the wall of afferent arteriole and pale staining lassus cells, which are important in transmission of impulses from macula densa to juxtaglomerular cells. And we have seen the role of juxtaglomerular apparatus in control of blood pressure. Other than the cortex medulla, in the cortex and medulla, you see the renal interstitium, which is nothing but connective tissue, which is less in cortex as compared to that of the medulla. So, this summarizes the histological details of the kidney in a histological section.